introduction that we need to talk about before we move into the third module. There's a lot actually in this slide. It's my last slide for this module, however, there's a lot here listed. All right, so our goal is to classify the organic macromolecules. There are 4 of them. They are the lipids, the proteins, carbohydrates, and nucleic acids. Okay? And when we talk about macromolecules, these are very large organic molecules, so they, for the most part, they have monomers and polymers. There is one exclusion that only has monomers. That would be, which one only has monomers? Lipids. Okay? So when we look at the chemical structure, the cellular function of these 4 macromolecules, they vary pretty significantly. I'm not going to verbally go into everything that the book has already done. You're going to see the questions, some questions on the online quiz that is due this Wednesday if you haven't already taken it, right? Some of you may have already completed it, but you do have a quiz on this chapter that's worth 16 points. It's on Canvas that you take as a quiz. It doesn't have anything on there saying, and I don't have any monitoring or anything like that, so as far as, the notes or books or information like that, I don't have anything, right? So I'm hoping you take full advantage and you get all 16 points, right? Unlike a lecture in class quiz, where it is all brain cells that you've retained, information you've retained. But if we look at our different macromolecules, we're going to start with the carbohydrates. The monomers are your simple sugars, right? So glucose is a pretty recognized simple sugar, one of a few. And then as we link these simple sugars up, we get what we call the disaccharides, as well as the polysaccharides, right? The disaccharides are just when 2 get linked together to create then another molecule, and then polysaccharides when we have many linked together. Carbohydrates are going to be very important for energy storage as well as structural material. We'll see the carbohydrate things such as cell wall structure coming into play as being carbohydrate based, right? When we look at plant cell wall structure, that is cellulose. Right, and we've heard that as far as us. So this should be stuff hopefully coming from, remember from your general biology. You have the glycogen storage is what we have for our cells as an energy storage. And then we do have in the bacterial world, starting in the next module as we move into cell structure of the bacteria, they too have a cell wall, and it is going to be composed of these carbohydrates. All right, and so we'll bring those guys up into play. Carbohydrates are generally under a molecular structure of, or formula of CH2O, right? Some sort of combination of this. So for instance, the sugar is glucose, which would be C6H12O6, okay? So you see that combination, that one, for every carbon you have 2 hydrogen, 1 oxygen. That's a pretty common formula for your carbohydrates. Yes.
When I was studying that specifically, doesn't it come with N? It's like C and H2O and big and this. And meaning the number can be different.
Well, N is just what they're assigning here as N is just the number they're saying. For instance, this, I'm saying it's 6 carbons in here, 6, so yes, this is basically 1, 1. So I'm timesing this by 6, this by 2, this by 6. It doesn't matter. It's a combination. It's a combination. N is just a placeholder for a number. That's all it means. So that is still a CH2O, right? In your case, you're just saying, instead of saying, you're just saying it's, you're saying CN and HN. All you're doing is putting a placeholder for that number. Okay, it's saying the same thing.
I just thought that I didn't know if the correct answer would be either the N in place or
Well,
the
let's
CH2O.
remind you guys again, these lectures, exams, are multiple choice, right? So I'm going to be providing you with a set of answers, and only one of them is going to be right. So, but no, I'm more familiar with this format. It's the same thing, literally the same thing. N is just a placeholder for the number. Now, let's see. We often see the carbonyls and the hydroxyls as functional groups in the carbohydrates. Okay, those are usually what we're going to see present. So when we look at our carbohydrates, you have your ketoses and your aldoses, right? Ketones and aldehydes. Most of your sugars, the carbohydrates, end in OSE. Not all, right? But many do. It basically rolls down to, if you see a word ending in OSE, you can make that leap that it's a carbohydrate, right? But not all carbohydrates end in OSE. Does that make sense? Okay. All right. Let's see. So, lipids. When we go through lipids, lipids are actually, they have no real set monomer, and they're actually a very diverse group. Within the lipids, of course, we have our phospholipids, very important for cell membrane structure. And we have our fatty acids that are attached to glycerol to create our triglycerides, right? Again, energy storage, so the fats. And then we have sterols, which are all your different steroids based. And then we have our waxy substances, our waxes, that are going to come into play in the lipids. So they are going to, these monomers are individual, and the monomers interact because lipids are hydrophobic. So they interact together due to that hydrophobic interaction, instead of bonding together to create polymers. Oh, and speaking of which, before let me just make a comment back to the carbohydrates. The bond that these monomers, when they link together, the bond that they form is a glycosidic linkage. Okay, so if you hear anything, any word that's glyco of some sort, you can automatically link carbohydrate. That's the type of molecule you're looking at. Since again, lipids aren't going to be bonding together, they're going to interact through hydrophobic interactions. However, that being said, we do have a very recognized energy storage molecule, which are the triglycerides. The triglycerides consist of the glycerol with 3 fatty acids attached, and those fatty acids are attached to the that glycerol through what we call ester bonds. The fatty acids can vary in length, as well as whether they have double bonded carbon, right? So that you can have what we call saturated fatty acids and unsaturated fatty acids. Saturated fatty acids are where all the carbons in that carbon skeleton for that fatty acid are single bonds. The unsaturated fatty acids are where we see the double bonds. And where you see a double bond between 2 carbons, it bends that molecule. It gives it a kind of an angle to it, making it less stackable. All right, looking at, so again, I'm doing brief overview for chemical structure, function, and the bonds as well as those monomers. So then looking at the proteins, of course, our monomers are the amino acids. There are about 20 amino acids that we see in the cellular, in the cellular system for the most part that are used regularly, right, to produce then your proteins. The proteins are the actual polymer. They're referred to as a polypeptide. When the amino acids link together, that is a peptide bond. So that's the bond formed when the amino acids link together. Amino acids are unique in the sense that, so that monomer is unique in the sense it has a central carbon. And then to that central carbon, it has an amine group, so NH3, and it has the carboxyl group. It has a hydrogen, and it has what we call an R group. All of your amino acids are identical with this portion, right? That portion is identical. Where they vary is the R group. That's what makes them the different 20 amino acids, makes them distinguishable from each other. When we have a bond form, that bond is going to be such that we will get what we call a backbone. So a polypeptide will have a backbone for amino. So you have your amino acid, right? 1, 2, 3, 4, 5, all linked together. The backbone for that amino acid is known for its fact that it follows a N-C-C pattern, right? N for the amino, C for the central carbon, C for the carboxyl, and then it'll repeat. The next amino acid will be N-C-C, and it keeps going. So that portion, this portion that's constant, ends up forming the backbone for that polypeptide. Okay, that's the constant. There is directionality to that structure, so we have a N-terminus, and we have a C-terminus. So it does have direction to it, okay? Now, proteins are really your workhorses for the cell. All the activity in the cell is performed by those. They play a role in transport, right? They play a role in structural support, such as, say, a cytoskeleton. Okay, across the board, everything, is dependent upon those proteins. And proteins have a very specific 3D structure. So proteins are consist of what we call, they have 4 levels of structure, right? The primary level of structure is the amino acid sequence itself, okay? Say, proline, isoleucine, leucine, glycine. That would be the first level of structure, so that primary level of structure, the amino acid sequence. And it's recognized by the fact that those peptide bonds are linking those amino acids together. So that's the bond form that's part of that primary level of structure. The next level of structure is referred to as the secondary level of structure. And the secondary level of structure has to do with the backbone interacting with the backbone of other areas on that polypeptide. So backbone interacting with other backbone. That can be a variable interaction, or it can be in very specific patterns called the alpha helixes and the beta pleated sheets. The bond that makes these particular 3D structures are referred to as hydrogen bonds. Okay, so the hydrogen bonds are going to play a role in making those alpha helix structure and the beta pleated sheets and the variable. Okay. And then we have the third level of structure. The third level of structure is also going to add to the 3D structure. Okay, it is not, the third level of structure, called the tertiary level, is not the 3D structure. Okay, it's not the 3D of the protein, which seems to be a misunderstanding or miscommunication when people are reading the text, right? It is another level of the 3D structure. Don't forget, the secondary is already added alpha helixes and beta pleated sheets. Those are 3D structures, okay? So the tertiary structure adds another level. In the tertiary structure though, the R groups are gonna come into play. So the R group will either bond with another R group or it will bond with the backbone. So it's either R group with R group or R group with backbone. And the interactions there, that bonding there, can vary. It can range from the strongest, which is the disulfide bridges, so that's a covalent bond, to an ionic bond. It can be a hydrogen bond. It can be hydrophobic interactions. Okay, all of those are type of interactions are and bonding can occur at the tertiary level, and that is again dealing with that R group. As long as the R group is in one, at least one of the parties is an R group, then it's considered that tertiary level. Now, all proteins have the primary, secondary, and tertiary level of 3D structure. The fourth level, which is the quaternary level of structure, only proteins that have multi-polypeptide components coming together are going to have that level, okay? So quaternary level is not required by all proteins, only those where multiple polypeptides are involved. And in that case then, those polypeptides are like subunits interacting together. And the interaction can be through hydrogen bonds, it can be through disulfide bridges, or ionic bonds, okay? So later this term, you're going to see, say, the structure for an antibody, okay? And it is actually going to be composed of 4, and it has sulfide bond, those disulfide bridges that are going to link them so that they are attached. Okay, I believe there's another one here. So, quaternary is not required by all proteins. They are the biggest workhorse, and they're important for structure, but probably the one thing that we recognize them for are the enzymes. The enzymes are the proteins that are involved in catalytic reactions. So when we get out of unit 1, and then we move into unit 2, and we start talking about metabolism, you're going to see the proteins pop up again, right? Because enzymes are going to play a big role in metabolism and promoting those reactions to move forward. Let me see if there's anything else I wanted to say on the proteins. I think that hit everything. All right, so then our fourth are the nucleic acids. The nucleic acids, we have the monomer, which is the nucleotide. It's not erasing very well. So, nucleic acids. Nucleic acids are going to be important for the genetic material, for cellular organisms, and we're going to bring this up again today, too, but for cellular organisms, the nucleic acid, that genetic material, is in the form of DNA, deoxyribonucleic acid. Okay? We also have RNA. RNA is a kind of an intermediary to get us to protein. So the genetic information, the DNA, stores the information, the directions on how to make the proteins, and then we will go through a process of transcription into RNA, and then using DNA as a template, and then we'll take the RNA. The RNA will be used as a template to make protein. Okay, we're not gonna go into all that right now, because it's part of the second unit. We will actually have that as part of the course. But so the nucleic acids themselves, right, the difference between DNA versus RNA is actually based on one location of the ribose sugar. That is the third carbon of that ribose sugar. Okay, in a deoxy, for the deoxyribose, it has just a hydrogen there. Okay, for ribose, it has a hydroxyl. Is that right? No. It's not the third. It's the second, because the third is always hydroxyl. Sorry. The nucleotides, when they link up, okay, so a nucleotide is going to consist of a pentose sugar, right? To it, we're going to have an OH at the 3' carbon. We're going to have, and the 5' carbon, we have a phosphate group. So we have a ribose or deoxyribose. So we have, I'm going to stick with pentose sugar, because we have a sugar. Okay, and then attached to that, of course, we have our nucleotide, our nitrogenous base. Nitrogenous base. Okay, now, nitrogenous base, so the pentose sugar and the phosphate, these remain constant. So when we have a macromolecule formed and we have the deoxyribose nucleic acid or the ribose nucleic acid form, this is going to be part of, right here, this is going to be part of the backbone. It's going to be constant. So you're going to have pentose sugar, right, phosphate, pentose sugar, phosphate, pentose sugar, phosphate. That's the backbone. Where the differences arise is in that nitrogenous base. And so we have 4 nitrogenous bases for DNA, and they would be what? Guanine, adenine, thymine, and cytosine. Okay? Oftentimes abbreviated to A, G, right, N, T, C. Adenine, guanine, these are your purines. Okay? How you remember that? There's a term, there's a little phrase, pure as gold. Pure, purines, as for the A, gold, the G. Okay? The pyrimidines are your thymine and your cytosine. When we have pairing occurring, what we call base pairing occurring, adenine will base pair with thymine, guanine will base pair with cytosine. This interaction between those nitrogenous bases, there's going to be a double bond between adenine and thymine, and not double bond, double hydrogen interaction between that hydrogen bond between adenine and thymine, and triple between guanine and cytosine. So 3 hydrogen bonds here, 2 hydrogen bonds here. The backbone does have directionality, okay? Just like we saw with proteins, those polypeptides, the polypeptides for protein had directionality. Nucleic acids also have directionality. They have the 5' phosphate side, and they have the 3' hydroxyl side. DNA is double-stranded. That means one strand will base pair with another strand. That's where this base pairing comes into play. And when they are base paired together, one strand is anti-parallel to the other. Okay? So if I have a sequence of DNA and I've got 5' on one side, 3' on the other, my paired, this is, our pairs are just linked together here, is gonna be 3' on the side of the 5', or it's gonna be linking to the side of the 5'. Okay, so it is anti-parallel. Parallel is that they both had the same 5' on both sides. They don't. They're anti-parallel. They're parallel when they base pair together, these two strands, to make your double-stranded DNA, right? But the 5' on one strand is matching with the 3' on the other. We will be going into this again in the second unit when we're actually talking about DNA replication, transcription, translation. Okay, here we're just kind of reminding you about that molecule. DNA is found double-stranded in cellular organisms. It is found, the RNA is found single-stranded, okay, in cellular organisms. That's the natural state. That being said, the nucleic acids are also the genetic information for viruses. So in this unit, we will also talk about the viruses, and viruses don't have the same requirements. Okay, viruses can be DNA-based or RNA-based. Cells are always DNA-based for their genetic information. Okay? The bond for between nucleotides, as you're reading through the material for this module, you might have noticed it had talked about that bond between the nucleotides being a phosphodiester bond. When two nucleotides link, they form that phosphodiester bond. All right. That covers macromolecules in a quick, brief overview. Again, it's just a repeat of what you went through in far more detail in general biology, and the reading is quite thorough. So make sure you're doing that. The reading. The quiz will be covering that same material too. Now, we're going to move into Module 3. I'm going to be covering Module 3 between today and Wednesday. Next week is the 4th module, so today is going to be the cellular structures, really. We're going to focus more on the prokaryotic side. However, the reading does include the eukaryotic side. So depending on the time, and depending upon the time, I may verbally talk you through that, but I know everyone had to take general biology. So, you know, going through and refreshing your memory just through the readings should be sufficient. So it'll just depend upon timing, okay, on my end as far as lecture time. I want to focus on the prokaryotic structure specifically for lecture because of the fact that that isn't covered as much in detail in the general biology class, right? In general biology, they stick with the eukaryotic. That being said, there's a lot of stuff that's very much the same or very, very similar, okay? So there may be a few terms that are new or structures that might be a little bit different. But overall, right, there's a lot of similar components. All right, so I'm not going to belabor this too much, but again, we do have a set of 9 learning objectives for this particular module to include the identifying common structural components of all cells, distinguishing prokaryotic and eukaryotic cells, and then identifying the structures of the bacterial cell at different levels. Basically, line item 3 all the way through 9 really have to do with the structures, whether internal, external, and what their functions are. Okay? So, we're talking about cellular organisms. So before we go too much into the cell structure, what is the characteristics of cellular organisms? One, we have to have growth. An organism must be able to grow, increase in size. Okay, not just in number, but they do actually increase in size. That can vary. Obviously, a multicellular organism increases in size more than, say, a single-celled organism. Single-celled organism generally increases in size solely as part of that cell replication process before they divide into two cells. Reproduction, of course, is very important if you're a cellular organism. Reproduction is very important. Reproduction is the increase in numbers, right? Now, for the purposes of a multicellular organism, it is the increase in numbers, not just, the increase in the, when we talk about reproduction, we're talking about the increase in numbers of organisms. If you're talking about a prokaryotic cell or any unicellular organism, we're really talking about the increase in number of cells, which are in themselves an organism, right? So it's not, cell division by default means you're increasing, you're reproducing, and you're increasing the number of cells. Okay, that's not the case with a multicellular organism, obviously. As the multicellular organism grows in size, they're going to increase in the number of cells, not necessarily the number of organisms. Responsiveness to react to environmental stimuli, right? So the ability to recognize the environment and respond accordingly. To metabolize and have these, take chemicals, right? Produce enzymes that promote the chemical reaction so that you can take one source of chemicals and change them in such a manner that they are now going to serve a function for that cell. And then, of course, we have, most importantly, cellular structure. And that main cellular structure that is required for a cellular organism would be a membrane. A cell membrane. The cell membrane defines what is inside the cell and what is outside of the cell. So when we get to talk about viruses, we're going to see that viruses aren't going to be able to meet the requirements of most of these. And therefore they are acellular. They are not acellular. They are not a cellular organism. They are acellular. They're not considered a cell. So that being said, what do all cells have? I listed one item right there, which is just a moment ago. A cell membrane, right? All cells have to have a cell membrane because the cell membrane defines it defines the cell. This is inside the cell, this is outside the cell. So it is the definition of a cell, the cell membrane. What else do they all have?
Genetic material.
All right, genetic material. Okay, in the form of, I'll put in the form of DNA. Oftentimes called chromosomes. Notice we didn't say where that was located though, right? We didn't say where that was located. We just said that they have to have genetic material. What else?
Cellular wall? Cell wall?
Nope, we don't have a cell wall. Cytosol or cytoplasm. Right, cytoplasm is that gelatinous stuff inside of the cell. All cells have that. What else?
Ribosome.
There we go. Ribosomes. Ribosomes. Very critical for the purposes of translating messenger RNA into protein, right? For protein synthesis. Anything else I think of? What about there? Okay. All cells have this stuff, whether it's prokaryotic or eukaryotic. It doesn't matter. Okay? Now, I need to just get rid of this. All right, so distinguishing between a prokaryotic and eukaryotic cell. What is the difference between a prokaryotic cell versus a eukaryotic cell? What does a eukaryotic cell have that a prokaryotic cell does not have? Membrane-bound organelles. One of which is the nucleus, right? The nucleus is just one of many membrane-bound organelles that a eukaryotic cell has that a prokaryotic cell does not. Okay? Of course, everyone always focuses on the nucleus, and I understand the reason why, but it is actually just a class of membrane-bound organelles. So that's the biggest difference. And why is that important for this cell? Well, by having that compartmentalization, by having those membrane-bound organelles that divide the cell into little rooms for different activities, a eukaryotic cell can be larger than a prokaryotic cell, anywhere from 10 to 100 times larger than a prokaryotic cell. Okay, so prokaryotic cells, the prokaryotic cells are going to include those organisms that fall in the domain of bacteria and archaea. They're more simple in structure. Okay, they don't have any membrane-bound organelles, including the nucleus, right? And they're typically around 1 micron in size. That's the average. Okay. Sometimes smaller, sometimes a little larger, but right around there is usually where they're at. A eukaryotic cell is going to be more complex because it has those membrane-bound organelles, one of which is the nucleus. It has that endomembrane system. You guys remember that? Where we talked about the proteins made into the endoplasmic reticulum, and then they travel to the endoplasmic reticulum to the Golgi apparatus, and then the Golgi apparatus to the cell membrane, where whatever's made is then deposited out, right, that endomembrane system. It also has, or can have, depending upon, let me rephrase that, it can have, depending upon the organism, 2 organelles that are energy producing, right? They are the
Mitochondria?
mitochondria is one of them. The other one being the chloroplast, right? The chloroplast, right? So the mitochondria and the chloroplast. And for us, animal cells, we don't have the chloroplast on it, because that's it. Chloroplasts are involved with light energy being converted into chemical energy, so photosynthesis, right? And mitochondria is chemical energy being converted into chemical energy, right? So it takes then, we're able to use then the energy stored in one type of macromolecule and convert it into a form that the cell can use, which that energy form is generally ATP. Right? Just as a quick side note, because I think you guys know this answer, but ATP, what class of macromolecule does it fall under? ATP.
I think it's the first one.
Nucleic acids. It is actually a monomer. ATP is adenosine triphosphate. It is a pentose sugar. It has a pentose sugar, 3 phosphates, and then the nitrogenous base being adenine. The pentose sugar, though, is ribose. So which nucleic acid is it most similar to? RNA. Right, it's RNA. Okay. All right. So we do have those 2 organelles that are found in a eukaryotic cell structure for energy conversion. In fact, as you move into this unit, They're going to talk about the endosymbionic theory. Has anybody started reading through module 3? Maybe, kinda, not really, not yet. We're just gonna talk about the endosymbionic theory, and that is the theory that the mitochondria and the chloroplast actually were prokaryotic cells that were intracellular parasites that lost the ability to survive outside of the host cell and kind of got downgraded from an organism to an organelle. Okay? Because their characteristics are very similar to a prokaryotic cell. In fact, as we move forward and we talk about mitochondria and chloroplasts, both of them have genetic information in them. So the genetic information for eukaryotic cells are going to be found in the nucleus, but there is also some information found in the chloroplast or mitochondria, depending upon if they have that particular organelle, because that particular organelle does have DNA within it. It supplies some of the proteins needed for that organelle, but not all. The rest of the information is located in the nucleus, right? So the organelle, you can't take the organelle out and have it survive and thrive on its own. It is definitely, it went in that hierarchy, it dropped down from an organism to a organelle because it lost that ability. Now for the eukaryotic cells, if we are looking at the cellular organisms for microbiology specifically, you're talking about the protozoans, algae, fungi, and of course, we do have a few animals, right, that we cover in microbiology. Not included in microbiology specifically would be the plants, right? But they are eukaryotic cells. All right. We want to then, I want to start from, we're going to look at a prokaryotic cell. We're going to start from the outside and move in. All right, and so you'll notice some of these learning modules might be mixed in a little bit just because of the order in which I want to cover the topic. So it may not actually be in the order as I have for the last module, or as we went 1, 2, 3, 4. Okay. But we are going to cover start at the outside going inward, and so as we start in the outside moving in for a prokaryotic cell, specifically a bacterial cell, although the archaea are very similar, you can have a glycolax. A glycolax is optional. A glycolax is similar to the intracellular matrix when you talk about, say, animal cells, okay? Maybe you don't recognize that term, maybe you do. It depends upon whether you've taken general biology recently or if you've recently taken A&P, right? So sometimes I try and pull the terms that you might recognize that might be familiar with the eukaryotic regards, in the eukaryotic regards, so you have something to base your,